بەڕێزانی لەسەر چاپتەری 8 سەکشنی 3 part 2 DNA replication. بەم پرسیارە دەست پێەکەین. How is replication error corrected? ئێمە دەرسی پێشوو باسی ئەومان کرد وتمان DNA replication usually occur with great accuracy. Only about one error occur for every billion pair nucleotide edit. يعني وتمان 10 power no. How is replication error corrected? How? We have three types of the DNA polymerase. One of the DNA polymerase called DNA polymerase repair or repair enzyme have repair function. So the fourth function of the DNA polymerase is that repair the mispairing nucleotide in the newly formed DNA strand. So the DNA polymerase have repair function that proofread mean determine the mistake and correct it. In the same way, friend may check a term paper for spelling errors. For example, if adenine paired with the cytosine, for example, adenine paired with cytosine. Adenine never paired with the cytosine, but if the DNA polymerase pair adenine with the cytosine, this is the mistake in the pairing between two nucleotides. DNA polymerase can repair the error by removing the mispairing cytosine. So which one is incorrect? This is the old strand. This is the mispair that paired with the adenine. The DNA polymerase remove this cytosine and it add thymine instead of the cytosine. So DNA polymerase prevent mutation in the newly formed DNA strand. What happens when the mistake of the DNA occurs? The base sequence, the base sequence is a single strand of the DNA. If we focus on the one strand of the DNA, this is called the base sequence. The base sequence of the newly formed DNA, not all strand, differ from the base sequence of the original DNA, molecule. So this is called mutation. So what is the mutation? Mutation, a change in the nucleotide sequence of a DNA molecule, is called a mutation. Mutation بعربي بيوتري الطفرة بكردي بيوتري بازدان is a change in the nucleotide sequence in a DNA molecule is called mutation. Mutation can have a serious effect. Some of the serious effect like cancer. For example, cancer, most of the cancer is due to the occurring of mutation in the DNA. Have a serious effect on the function of an important gene. We have 25,000 of important genes in our human genome. These genes are important for our life, our survival. If the mutation occur from any gene, may cause disease or disorder. Sometimes the genetic disease is called genetic disorder. Most of the genetic disorder caused by mutation and disrupt an important cell function. Right factor that cause the mutation or mutagenic substances. Mutagenic substances include some error escape the repair. If DNA repair repair enzyme does not find the mistake. So the mistake escape the repair. This is called mutation. Chemical substances like air pollution, preserve material that has been added into some food, and other substances in the chemical nature. Ultraviolet radiation or radiation, for example UV, some of the ultraviolet radiation that come from the sun can damage the DNA. Why DNA repair enzyme important to an organism survives? DNA repair enzyme, see this term, DNA repair enzyme mean DNA polymerase. DNA polymerase repair. Important for organism survive. همو ما نزانین که اگر DNA polymerase enzyme نبی mutation روبا. اگر mutation روبا او کاتا چی رویا؟ لانه cancer روبا. Because if the error escape repair, a change in the nucleotide, a change in the nucleotide sequence of a DNA molecule will occur. So what's the meaning? The nucleotide sequence of the DNA molecule change, meaning mutation, and some mutation can lead to cancer. DNA replication is an elegant process in which genetic information is passed from one cell for the thousands of the generation. Emma Zaninka, during embryonic formation, one of the cell, for example, contain 46 chromosome. This 46 chromosome is called diploid 2n. Ama naoning zygote. This zygote by DNA replication. This zygote is diploid on 2n zygote. Forms billion of cells. Of the human. See this figure. This is one of the cell. This is nucleus of the cell. Contain 46 of the chromosome or diploid 2n. This is zygote. By DNA replication form million or even billion of cell, and each cell contain 46 of the chromosome, by which process preserve this number for all cells by DNA replication. So the DNA replication, by DNA replication, genetic material pass from one cell to thousands of the generation, and it also explains how mutation can arise and lead to altering the cell and organism. یەکێک لە ئیسفادەکانی ئەوەی کە ئێمە DNA replication ئەخوێنین ئەوەیە کە ئەزانین mutation چەندێک خەتەرە بۆی بزانین چۆن prevenتی mutation بکەین. Note number 2. DNA replication is one way to understanding and treating various types of the human cancer. So benefit of the DNA replication, study of the DNA replication help human to find treatment and prevent various types of the human cancer. Mutation have advantage and disadvantage. Some mutation advantage for the cell. The change allow the individual to survive and reproduce better. So sometimes mutation allow some organism in some place to survive and reproduce better. So these variation increase the population over many generations. طبعا لسال 1960 كان مادي كان بكار هنا وبناوي DDT. أما دي insecticide بو. This insecticide kill all insect in the agriculture. After past of some decades, these insects do not kill by this pesticide or insecticide because mutation allow the insect to prevent themselves from these substances. So mutation can cause the organism to survive and reproduce better. بس سيري mutation كان ل HIV أو كلا chapter سياخ من دوتانا. HIV have RNA. This RNA mutate, and this mutation able to HIV to recognize other cell, other co-receptor in the other cell like helper T cell. So coronavirus شروعا coronavirus زور mutation كا زور بي زوري زانا كان بروان ويا أو coronavirus أو كلا تشاينا هبو زور فرق هي أو إلى إيران أو إلى إيطاليا أو إيش إلى كردستان. أما mutation طبعا بوان إسفادي هي mutation بوان إسفادي هي بس بو human خرابة. Sometimes mutation that are not repaired أجر mutation باش نكريتو چاك نكريتو can cause disease such as cancer. Mutation that are not repaired can cause cancer. Various types of the cancer is caused by mutation. One of the mutation that occur in the gene that controls cell divide. We have a gene in all human cells called gene for cell division, gene how cell divide. If this gene is mutated, so the cell will divide repeatedly, continuously, without stop. This is called cancer. Uncontrolling of cell division is called cancer. So if the gene that controls cell division mutate and the cell divide continuously without stop. Explain how a mutation during replication can affect a protein that is synthesized. إما إن شاء الله أول chapterي هشت إخواني لchapterي هشت سكشني شوار يعني دوام سكشنا أما بس بكرتي بوتام باسكم أجر داوي لبردناكم ليش تاني قشتن لسكشني شوار جوي لي بقرنيان سيري بكن متابعي بكن تيكن إن شاء الله. خوي DNA بيسكوينسي هي واتا سكوينسي كي تايبتي هي اوردري كي نيتروجينز بيسي تايبتي هي ام اوردرا بلين كوبي ابي ترانسكريبت ابي اتشبو بلين mRNA كودي هي او كودانا ابي بامينو اسيد سكوينس كانشي اوردركي وتايبكي ديترميني بلين شيبي بروتينك شيبي بروتينكش ديترميني فانكشنك يكات استا لم رسمه ابو تعالونا كمان برزان سيري ام رسمه بكن. This is the DNA. اما DNA consists of the double strand and wrap around each other. This process by mean mRNA is formed, mRNA called messenger RNA formed from the one of the strand of the DNA called transcription. Transcription يعني استنساخ. وكو تو شون كتابك هي استنساخك يقلا بري استنساخك بشوية. This is called one gene. This is called one gene. This gene, one of the strand of the DNA, acts as a template for formation of the mRNA. All of the information in the DNA sequence transfer to the mRNA without changing any nucleotide in the DNA or original DNA. This is called transcription. We will mention in chapter 8 section 4. By enzyme called RNA polymerase. RNA polymerase are enzyme or is enzyme that catalyze formation of the mRNA from the DNA template. After that, this mRNA go out to to the from the nucleus to the cytoplasm, then link to the ribosome, form amino acid sequence. This is called amino acid sequence. This is called polypeptide. Polypeptide, you can say protein. Polypeptide. Polypeptide consists of the many amino acid. This is amino acid linked together by peptide bond. So genetic instruction transfer to the polypeptide via mRNA. This is nucleus. Transcription occurs in the nucleus, then form mRNA. mRNA goes to the cytoplasm, linked to the ribosome to form a specific protein. This polypeptide have specific function, have specific shape before the function. Have specific shape. This shape determine their function. The shape of the protein determine what? The shape of the protein determine determine their function. So what is the effect of the mutation? If the mutation occur here, for example, ultraviolet from the sun cause the damage to the DNA. This damage of the DNA transfer to the mRNA. So mutated DNA form mutated mRNA. When the mutated mRNA translate, form abnormal polypeptide. Abnormal polypeptide mean change shape. The shape of the protein if change form abnormal protein cause the genetic disorder. The shape of the protein determined by three things: number of the amino acid, type of amino acid, and order of the amino acid. Type and number of order of amino acid determined by the mRNA. mRNA is determined by the DNA. So if the mutated DNA here transcripted to the mRNA, the mRNA is incorrect. The incorrect mRNA can result in correct amino acid according to type of amino acid, number of amino acid, and order of amino acid. When the type and number and order of the amino acid in a specific polypeptide are changed, so it affects their shape. The shape of the protein determines function. كيلبيريتي antibody شكلكي چونه؟ بالتأكيد Y shaped. This Y shaped is determined by type, number and order of amino acid. Order, type, number of amino acid is determined by mRNA. mRNA is determined by the DNA. If the DNA is mutated, the mRNA is incorrect. If the mRNA is incorrect, so either type, number or order of the amino acid has been changed in the polypeptide. If the polypeptide change, their shape is changed. If the shape is changed, so the protein unable to perform normal function. Skin cancer can result of a DNA mutation. One of the types of the cancer is called skin cancer, result from the DNA mutation. Skin cell receive too much ultraviolet radiation. اگر skin ultraviolet radiation زور برکه اوکاتا ابیته هوی mutation. اینە سەر تایپەکانی mutation. بەڕێزان ئەمە ڕێک چاپتەر 9 سێکشنی 1 چاپتەر 9 سێکشنی 1 جا ئەگەر چاپتەر 9 تەرک بوو ئەوە ئەم بەشە هەتا خوارەوە تەرکە. ئەگەر تەرک نەبوو ئەوە ئەمە سەر بە چاپتەر 9 و وە لێرە ئەیخوێنین. باشە بەڕێزان ئەگەر چاپتەر 8 داخل بوو بەس چاپتەر 8 داخل بوو چاپتەر 9 داخل نەبوو ئەوە ئیتر لێرەوە داخل نییە بڵین تاوەکو سێکشنی سێ تاوە بێ بەڵام سێکشنی 4 داخڵە. بەڵام ئەگەر چاپتەر 9 داخل بوو ئەوە ئەمەش داخڵە. یەکێک لە تایپەکانی mutation بریتێ لە germ cell mutation. germ cell mutation and somatic cell mutation بەم دیفرەنسە ئێمە ڕوونی ئەکەینەوە هیوادارم بەدڵتان بێ. Germ cell mutation occur in the gamete. So this is the keyword. Keyword for the germ cell mutation. The germ cell mutation occur in gamete, either in sperm and egg. While somatic meaning body, occur in the body cell, like skin cell, like blood, like connective tissue, like epithelial tissue. All of these types of the mutation occur in our body except gamete is called somatic cell mutation. The germ cell mutation do not affect the organism itself. بونمونه اگر a male have mutation in his gamete, so do not affect the male itself, but this male transfer abnormal gamete to his offspring. But the somatic cell mutation can affect the organism itself, but they do not affect the offspring, cannot be inherited, while germ cell mutation can pass on to offspring. The third type of the differences, they can be passed on to offspring, but somatic cell mutation cannot be inherited. هنا سر بابتي كأيش chromosomal mutation. Chromosomal mutation is very important. We have two main types of the chromosomal mutation in the structure and in the number. In the structure and in the number. Chromosomal mutation have two main types. The type number 1 in the structure, type number 2 in the number. In the structure include deletion, some part of the chromosome delete in a specific region, inversion in a specific segment of the chromosome, and translocation, transfer specific segment of the chromosome from one homologous chromosome to another non-homologous chromosome. While in the number we have two types of the chromosome in the level of the number. The first one in the level of the gain of the chromosome, mean every human have 46 of the chromosome, but when the number of the chromosome increase 1, 2 or 3, this is called gain of the chromosome. Chromosomal mutation in the level of the gain, like Down syndrome. Down syndrome have 47 of the chromosome and has an extra copy of the chromosome number 21st. This is called chromosomal mutation in the number. While loss of the chromosome, like 45 chromosome, some person have 45 chromosome. This is called Turner syndrome. This is not include for ministry exam question. Turner syndrome. Turner syndrome have she-male and have characteristic of the she-male hermaphrodite. This Turner syndrome have 45 chromosome instead of the 46 of the chromosome. Chromosomal mutation involves change in the structure of a chromosome or lose or gain of the chromosome. This is types of the chromosomal mutation. The first one of the chromosomal mutation is deletion. Deletion is the loss of a piece of a chromosome due to breakage. The breakage causes the chromosome breakdown and separate a small pieces of the chromosome, like this figure. This figure is called deletion of small pieces of this chromosome. This piece of the chromosome deleted by breakage, then decompose in the cytosol and become chromosomal mutation. The second one is called inversion. Inversion. The inversion, a chromosome segment break off. The first step is break off, flip around, then flip around backward and then reattach. Flip around backward, then reattach. See the second figure. This chromosome is one homologous chromosome. The orange part located up of the chromosome and the red part located below the red. This is called centromere. This chromosome, the first step right down here, break off, right down here, flips around backward, then reattaches. So the resultant of this chromosome, the red located up of the chromosome, while orange part located down of the chromosome. But the original chromosome, orange located in the up of the chromosome, while the red part located down of the chromosome. This is called inversion. Inversion. See this chromosome. This chromosome arranged in the following gene. The gene represented by A and another gene represented by B, C, D, E, F. This chromosome is one of the single chromosome of the homologous chromosome. برزان سيري أو رسمة بكان أما كروموسومي كأما طبعا جيني تسلسلكي بمشيوية A, B, C بلام ليرة أما سنتروميرة جيني تانية باشان D, E, F بريك أوفا به أقليرة بريك أوفا به رك ليريشا بريك أوفا به باشا كبريك أوفا به أما سيبريتا به باشان flips around. Then the first step is break off. This is the break off of the chromosome in two specific regions. Then second step is flip around واتا أخلته كخلايا شي رؤيا then reattach أما fixta أو هيك هلا سرا أم إيديا هي كوبو هي إيري هاتوتا سرا وأم سيبيا هي خواريا هاتوتا سروا اما بيوتري reattached لرا ام ستيب اخوتان بينوسن اما third step ابي باتشي reattaches. So inversion, a chromosome segment break off, first step break off, flip around backward and reattaches. And third types of the chromosomal mutation in the level of the structure, a piece of the homologous chromosome, one chromosome break off and reattach to non-homologous chromosome. So the condition of these types of the mutation takes place between two homologous chromosomes. For example, this is chromosome number, for example 13. Transfer a specific gene here, this specific break and transfer to non-homologous chromosome. For example, this is another chromosome number, for example 17. This is two non-homologous chromosome. This is one homologous chromosome and this is another homologous chromosome. Transfer of the genetic pieces between two non-homologous chromosomes is called translocation mutation. Then how about the non-disjunction? This is the illustration of the meiosis. During meiosis or phase meiotic division or phase meiosis, homologous chromosomes separate from each other and migrate to each cells. During the second meiosis, the sister chromatids separate from each other and move to each cells. This is separation of sister chromatid. Sister chromatids separate from each other and form 4 haploid normal cell. Each cell contains 23 chromosomes. This is represent one of the homologous chromosome. Meiosis is responsible for producing 4 haploid cells from one original diploid parent cell. Failure of chromosome separation from one another during either round of meiosis result in non-disjunction. This can occur either in meiosis 1 or meiosis 2. This is separation of sister chromatid from each other in the form of normal, while these 2 chromatids do not separate from each other, and both of the sister chromatid goes to one cell. This is called non-disjunction. Normal chromosome separate begin with the diploid cell at the start of meiosis. We will focus on pair of chromosome as they segregate. This pair can represent either autosome or sex chromosome. We have 23 chromosomes in each cell. 22 of the chromosome called autosome. The last pair is called sex chromosome. In male contain XY chromosome, while in female contain XX chromosome. So from the first chromosome to 22 chromosome is called autosome. This figure represents one homologous chromosome. This is one homologous chromosome. Each homologous chromosome consists of 4 chromatids. This is 2 sister chromatids that are joined together by a specific structure called centromere. Centromere do not contain any gene. So normal chromosome separate during these homologous chromosomes separate from each other during meiosis 1, while sister chromatids separate from each other during meiosis 2. During the phase meiosis, homologous chromosomes separate, that's shown in the movie. So homologous chromosomes separate from each other. The end result in 4 haploid cells, in each with one sister chromatid, now is called chromosome. So from the second meiotic division or meiosis 2, sister chromatids separate from each other. How? From here, this is called centromere, split, then the sister chromatids separate from each other. So you must focus on the first meiotic division. First meiotic division, homologous chromosomes separate, and the second meiosis, sister chromatids separate from each other. This is normal chromosome segregation in meiosis. This is called chromosome, single chromosome. In animal, these 4 haploid cells are called gamete. These 4 haploid cells are gamete. When they fuse with the normal gamete from opposite sex, normal diploid cells are produced. This is diploid cell. Non-disjunction occur either in meiosis 1 and meiosis 2. Now we first focus on non-disjunction in meiosis 1. If the homologous chromosome, this is homologous chromosome, fail to separate, then one cell produced with both homologous chromosome and the other cell has no copy of this chromosome. In the other cell, other two cells produce from one cell. There is no copy of this chromosome instead of the usual one, while these two cells contain extra copy of chromosome. For example, this chromosome is 21st. This chromosome is 21st chromosome. This cell do not contain any copy of the 21st chromosome. This cell contain two extra copy of the 21st chromosome. Then produce 4 haploid abnormal cell. 2 of them contain no copy of 21st chromosome, and other 2 contain extra copy of 21st chromosome. This is 4 haploid gamete. 2 of them contain extra copy of 21st chromosome, and other 2 contain no copy of that chromosome. All of these cell produced are abnormal for this chromosome. When normal gamete of male fuse with these abnormal, these 2 zygote contain 3 extra copy of the 21st chromosome. This is called trisomy. When the other cells that do not contain any copy of 21st chromosome fuse with the normal gamete, what will produce? Produce monosomy. Monosomy isn't include. So trisomy, for example, this zygote contain 3 copy of 21st chromosome, like in Down syndrome. During meiosis II, also non-disjunction occur. Let's observe what happened when the non-disjunction occurred during the second meiotic division. Homologous chromosomes separate normally, and each of which goes to one cell. If the sister chromatid of this sister chromatids fail to separate from its sister, so it is called non-disjunction. One cell, then 2 abnormal cells are produced. One having 2 copies of this chromosome, and other having none. And 2 normal gametes are produced that contain normal chromosome. So from the second types of the non-disjunction that occur in the meiosis II form 4 gametes. 2 of them are normal and 2 other are abnormal. One of the abnormal contain 2 copy of the 21st chromosome, and another contain no copy of the 21st chromosome. This is called non-disjunction. When this cell that contain 2 copy of the chromosome fuse with the normal gamete, also contain 1 copy of the 21st chromosome, and this is contain 2 copies of the 21st chromosome. What will produce? Produce trisomy 21st or Down syndrome. برزان استينا صار gene mutation. باسی non-disjunction ماكن non-disjunction. A chromosome failed to separate from its homologous chromosome during meiosis. Meiosis 1 كلا فيديو كيش باس مانكرت and meiosis 2. One gamete receive extra copy of the chromosome, for example chromosome number 21st, and another no copy. An example of the non-disjunction result in Down syndrome. Down syndrome sometimes is called trisomy 21st. Trisomy 21st. Trisomy 21st is same as Down syndrome. Now we explain gene mutation. Gene mutation have many types. One of the types is called point mutation. The point mutation have 3 types: substitution, addition, remove of a single nucleotide. Just one nucleotide is add or substitute or remove is called point mutation, which is a change occur within a single of gene or other segment of the DNA of the chromosome. We have one of the types of the gene mutation or point mutation is substitution. See this sentence: the cat ate the fat rat. The cat ate I ate the fat rat. This is the normal sentence, contain many letters and words. In the substitution, one nucleotide, just one nucleotide replace another. And this substitution occur, if this substitution occur in a codon mean in the mRNA, the amino acid can be changed. Some notes about the substitution: only one nucleotide change, only one code is changed, only one amino acid change. The number of the nucleotide in the DNA is not gain or loss. So how many letters exist in here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. And this normal, this is normal. The cat ate the fat rat. One nucleotide, one letter replaced by another. So for example, this C letter replaced by J. The cat ate the fat rat. So this is called mutated. How many letters exist here? Have the same letter that has been exist in the normal sentence. What is the deletion? In the deletion, one nucleotide is removed from the normal. The cat ate the fat rat. When the C letter, when this C is letter removed, the nucleotide shift, because the nucleotide arranged in 3 adjacent nucleotide, which is called codon. The codon consists of 3 nucleotides. When this C is removed, this A come to here. So all of the codons down the mutation has been changed. So this sentence is abnormal. Characteristic of the deletion: one nucleotide is deleted or removed. Many codons وثمان codons چیه؟ 3 nitrogenous base codon او كون رقمي اساسي لو كورك 11 رقم اگر یک رقم ديليت كيتر ورنا جيرا رقم كبود nitrogenous base كان سي ابن بكود ويان codons. Many codon have been changed. اگر هر کودونی کیش بو امینو اسیدکه، هر یک کودونو بو امینو اسیدکه. اما بزن سه nitrogenous base پیانو تره کودون، هر کودونی کیش بو امینو اسیده. So many amino acid in a polypeptide is changed. ماشا كواتا اینجا have disastrous effect on proteins shape and function. آخر خال deletion of the nucleotide called Frameshift Mutation, because many nucleotide has been changed. So deletion mutation, one or more nucleotides in a gene are lost. This loss can cause incorrect grouping of remaining codons. بس ما group من codon, 3 nitrogenous base is called codon. Each codon code for one amino acid. This is called frameshift mutation, making all amino acid downstream change. In turn, can have a disastrous effect on the protein's function. Another types of the frameshift mutation, one or more nucleotide is added, lera lost, balam lera added, which can result in frameshift mutation. This insertion can cause incorrect grouping of the remaining codon called frameshift mutation, making all amino acid downstream change. In turn, can have a disastrous effects on the proteins. In insertion mutation, this is a types of the frameshift mutation. The cat ate the rat, the fat rat. This is normal. When the G letter is added into the group, all of the nucleotides shift, and this is called downstream of mutation. G is added, then C, A, this T shift into the another code, then this E is shift to the another code, this E also another code, and all of the nucleotide or all of the codon is changed downstream. Characteristic of the insertion: one nucleotide is added, many codons have been changed, and also many amino acid in a polypeptide is changed, and have disastrous effect on the protein's shape and function. And this is a types of the mutation, frameshift mutation. سيرس مكب كان أما طبعا إيك يكلا تايبكاني point mutation substitution. This T is removed, and instead of it insert a adenine into this. When the normal DNA transcript to mRNA produce glutamic acid, this is normal protein, normal amino acid, normal codon, normal DNA. While during mutation, the second nitrogenous base A replace T, the code, this code is changed from GAA to GUA, then the amino acid changed to valine. This is called substitution. In the normal, this is example from frameshift mutation type of deletion. This is normal DNA. When this normal DNA transcript to mRNA, this transcription of the mRNA then translate to amino acid. The first amino acid must be cysteine, second amino acid glutamic acid, third amino acid glutamine, normal 3 codons, normal 3 amino acid sequence, normal DNA. While when the G or guanine is removed from the DNA sequence, the mRNA has been changed. 3 different codons form 3 different amino acid. Cysteine changed to tryptophan, glutamic acid changed to serine, glutamine changed to arginine. So this polypeptide contain abnormal amino acid than the normal amino acid. Type of the amino acid, number of the amino acid, order of the amino acid is very important for the shape of the protein. The shape of the protein determines their function. So this is called frameshift mutation type of deletion. Thank you so much. Goodbye.